All right, hey everyone. We just finished up class and I wanted to do a nice little review about translation, ask you some questions and do a little bit of in-depth talk about translation. So I have a question for you and please feel free to follow along and try to solve this as I am talking about it. Yes, so using a codon table, specifically this one here, feel free to come back and pause to look at the table. There was definitely a better way to do this, but I did not do it. Using the codon table, what amino acid sequence would result from the following complementary strand of DNA? Not the template strand, the complementary strand. So here's the complementary strand. And where do we find a complementary strand of DNA? So, we have our DNA double helix, very nice. And usually we're going to, in the case of our not replicated. In the case of transcription, we usually have a specific gene, a specific length of DNA that we want to transcribe because it has some specific information that we want to maybe turn into a protein, or we want to regulate another gene further down the line. So that information is on one of the strands, because if this was like AAT, and that's what we wanted, this one would be TAA. And if we wanted the instructions for AAT, then we could only use this strand and not this strand. And the one that we want to use, we call it the template, because we're going to use it as a template to make our RNA that will then turn into a protein maybe. So the question is asking, if we wanted to make such an amino acid sequence, we're given this complementary strand of DNA. And so we're going to use a template strand of DNA, and we're going to translate it. Or no, excuse me, we're going to transcribe it. Forgive me, and I will say something about that in just a second. We're going to take our template strand of DNA, and we're going to transcribe it into an mRNA sequence. We are then going to translate that into a polypeptide sequence, also known as an amino acid polymer. Wait, I don't know how to spell. And polypeptides are also what we call proteins. So it's always a bit difficult to keep transcription versus translation straight in our heads. Like you could be asked an example, a question on an exam, like, what is the goal of transcription? And I've seen people in the past say, oh, in transcription, we do this and this, but they're actually referring to translation. And, we don't want to do that. So the way that I keep transcription in mind, which is the taking DNA and making RNA. Transcription, I think of someone like copying a book. And, some people have different dialects for their languages. Like you could live in Northern Germany and you'd speak one aspect or one dialect of German, and you could live in the east of Germany and you'd speak Bavarian. So think of like a monk, a German monk, and he's reading a book, because our DNA is a lot like letters in a book. And this monk is sitting here and he wants to rewrite this book. He wants to copy it into his dialect of German. So he's going to take this familiar language. It's still German, it's still Deutsch, but he wants to translate it into a different flavor of German. And that's what transcription is. We're still writing in the same language using our nucleotide bases, our A's, T's, C's, and G's. I don't know why the pen is doing this. It's being kind of mean. How rude. Our A's, T's, C's, and G's. But this time, when we transcribe it, we want to put it into our different dialect. So we're going to use the same language, but we're going to use our own dialect. So we're staying in the same language. We're still using our nucleotide bases, but we're going to turn it into a different flavor because we start with DNA, and all of our molecular machinery relies on mRNA to go into our ribosomes to be transcribed, or excuse me, translated into proteins. So transcription is, we're still using the same language, the same nucleotides, but we are using a different dialect, a different flavor. We're going from DNA to RNA. Translation, however, we're starting to use a different language. Maybe this monk, he transcribed his book. Oh my God, this is ridiculous. He transcribed his book into his Bavarian dialect of German, but then an Italian monk comes in and he wants to translate the book into Italian. So he's going to use a different language. He's going to translate it into his language. And this book was written in nucleotides, our nucleotide bases, our A's, U's, C's, and G's. But this new language, we're not using our nucleotides anymore. We're using more complex language. Not that German is less complex than Italian, but we're using proteins. We're using amino acid polymers. We're no longer writing in nucleotides, but we're writing in amino acids that get strung together and maybe folded up in weird ways to make proteins. And that is what translation is. We're changing our language. We're no longer using nucleotides, we're using proteins. So that's how you can keep those straight. So that's what we're doing. And we have, wait, that's the pencil. So we have our complementary strand. And remember, we use the template strand to do this business. But we're given the complementary strand. And the complementary strand, thankfully, is just like the opposite of our template strand. As we drew on our DNA molecule before. And we know that the nucleotides, oh my God, it's ridiculous. The nucleotides, sorry, line up. They complement each other. If this is the template strand, then we know that the complementary strand is going to be, opposite, complementary to our template strand. So if we want to end up making an amino acid sequence, we need to start with the template strand. And so we need to convert this complementary strand and find the complement of it so that we can have our template strand of DNA. So we want to make amino acids, but we know that we can only do that with the template strand. So we first have to convert this complementary strand here into our template strand. And let's pay attention to the direction. And if you haven't, you probably haven't, but let's see, where are we? I did a pretty lengthy review of all of the most important parts of DNA replication this last Sunday in our study group, which anyone is welcome to join, by the way, and the link is posted. But if you go into LA Resources, which is where you'll find all LA stuff that we make, and exam through material, in this, we talk a lot about the directionality of DNA, what the 3', 5, and 5' means, what it means for replication specifically, which way enzymes can read our DNA and which way new complementary DNA is built. And in that talk, we talked a lot about how, oh wait, where'd it go? I'll just do it again. Of course I did that. Hopefully this is still recording. I turned the screen off instead of taking a screenshot. Very cool. Let's try that one more time, or maybe I could even find it here. Yes, maybe not. Okay, here we are. So in that review, we talked extensively because it's very important. And if you do not have this down, you should definitely practice. The fact that when we have DNA, let's draw our nice familiar replication bubble. Let's draw a more neat replication bubble. That was even worse, but okay. We have our origin of replication, and we have our template strand of DNA. Remember, this is replication, so we have more than one template strand. So keep that in mind. We're not doing transcription. So the template strands, and then we're going to be synthesizing a new complementary strand of DNA that is, opposite, complementary to that one. So temp and then complementary. And our enzymes, they attach to our template strand, and they can only read our template strand of DNA. And keep this in mind for all of this molecular genetics that we're doing for transcription, translation, and replication, that enzymes read template strands of DNA from the 3' to the 5' direction. So this would be the 3', and this would be the 5'. And just like we read like a number line, 1, 2, 3, 4, 5, we read 3, then 4, then 5. Our enzymes also read from low number to high number. So if we're looking at this template strand of DNA and we're told that 3' is over here and 5' is over here, we know that our enzymes are going to be moving this way. And over here, where the replication fork hasn't gone yet, we have our still double-stranded DNA. We know that this strand here is going to, I'm going to try to not use that color. This strand becomes this strand, and this strand becomes this strand. So this strand and this strand, this template strand and this template strand are complementary to each other because they used to be connected double helix here. And if this were an A, this would be a T. And so if they're complementary to each other, and we can also call that being anti-parallel, if 3' is here and 5' is here, then we know that 3' for this bottom template strand would be here, and the 5' for the bottom template strand would be here. So which direction would our enzymes go? We know that enzymes read from low, they read our template strands of DNA from 3 to 5. So our enzymes, for example, our DNA polymerase 3, it would be moving this way because it reads from 3 to 5. And I guess I could draw it again, but so we have, if you were to zoom way, way, way in on this DNA, we would remember or recall that a DNA molecule, it's hard drawing this sideways, is it looks a bit like this. And we have this guy and this guy. And we have our 1 carbon here, 2 carbon here, 3 carbon here, which is important. So we call it the 3'. Our 4th carbon is right here, and our 5th carbon is right here. So we call this one 5'. And so we kind of have a directionality to our DNA. Once again, this end, so if you think of the molecule as having like 2 ends, this end over here is closest to the 3rd carbon, so we call it the 3' end. And this end over here is closest to the 5' carbon. It's over here, so we call it the 5'. And so we like to say that the entire strand is 3 to 5 because every single nucleotide along the way is oriented from this 3 to 5 direction. So keep that in mind. And okay, where is I going with this? Oh yeah, Complementary strands. Complementary strands are anti-parallel to each other. So let's forget about replication for now and go back to our problem. That was just a nice little review in case you don't want to watch the video. So complementary strand, we know it's going to be anti-parallel to our template strand. And remember, it's the template strand. I should try to be color consistent. It's the template strand that we use to transcribe and translate stuff for the future. So if 5' is over here, then on our if 5' for the complementary strand is on this left side here, then the left side of the template strand would be 3' because they're anti-parallel, they run in opposite directions. So this complementary strand would be going this way, and the template strand would be going this way. So 5'. So what is the opposite of A? I'm just going to crank this out. Hopefully we know our base pairing rules by now. Keeping in mind that this is DNA. So we have T, A, C, G, T, 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 A, C, A, C, T. And this would be our 5' end. Oops, I said 5. That looked like a funky letter. So this is the template strand of DNA. This is what that would be if we were given this complementary strand. And now that we have a template strand of DNA, we can use this for the next part of our process. And remember, what do we do with DNA if we want to convert? Like eventually, we want to make an amino acid sequence, like the question was asking. So what do we want to do then? We want to transcribe it into RNA. And remember, transcribing, we're just changing the flavor of our language. We're using a different dialect. And because we're still using the same language, we are sticking with our nucleotide base pairs. So we're going to transcribe this into an mRNA sequence. And this is also going to be complementary. Should I draw it? No, I'm not going to draw it. Remember your transcription. And the mRNA product of transcription is also going to be anti-parallel or complementary to the template strand. So if the left side of our template strand was 3', then the right side of our mRNA strand is going to be 5'. You can see how we're switching back and forth and back. That's how our enzymes have to work because of the way that polymerases are able to add new nucleotides to a sequence of either DNA or RNA. It's just how they work. So let's transcribe this. Let's convert this DNA sequence into an RNA sequence. Remember, RNA, we don't use thymines, we use uracils instead, and the difference is a single methyl group. So let's do that really fast. We know this would be ATG. That's not right, sorry. I messed up immediately, very cool. Even after I said it, AUG. Da, 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 da. So how's your day going? This would be UGA. And this would be 3', remember anti-parallel. So we have our mRNA, very cool. And now this is what we talked about in class today, translation. And translation, as we articulated a bit earlier, is, and just like it sounds, it's all about changing the language. So we're no longer using our nucleotide bases anymore, color consistency, Gerber. We're no longer using our nucleotide bases anymore. We are now starting to use individual amino acids. So we're using a different language, we are translating. And this is, oh, by the way, wait, how should I do this? Yeah, so before we talked about it in class today. We have this mRNA after it's been processed, given its poly A tail, its GCAP, it's had its introns cut out, maybe some other stuff that we will ask about in this question later. We're going to have that mRNA strand. Why is the pen doing this? We're going to have this mRNA strand. And what's going to happen is that it's going to be shuttled out of the nucleus. And it's going to be recognized by our ribosomes in our cells. That's what ribosomes are important for. You learn about it in your cell biology. We have a bunch of ribosomes floating around in our cytoplasm. There are also some embedded in the membrane of our rough endoplasmic reticulum. That's why the endoplasmic reticulum is called rough sometimes, because it has little ribosomes embedded in it. So it looks like it would feel rough, and so our mRNA is going to be recognized by a ribosome. And ribosomes in eukaryotes, they're different size than in prokaryotes. I don't think you necessarily have to worry about it. If you take micro, you'll learn about it. But our ribosomes typically have two parts. They're made up of two subunits that make up the whole ribosomal unit. We have obviously the large subunit and the small subunit. And the small subunit is going to be the one that actually recognizes this mRNA sequence. And it's going to attach to it. And then that's going to invite the large subunit to come and sit on top. And with the large subunit, let me redraw that. It's going to invite the large subunit to come and sit on top. And with the large subunit, we then, that brings with it some important players in our translation. I'm trying to figure out what colors I should use. So we have these special things, these special kinds of RNA that are called tRNAs. And tRNAs are special little loops that have some special business going on that look like a cactus. And these RNAs are, they have RNA, and so they have their own nucleotide sequences. What's really important about these tRNAs is that they have something attached. They have these little things, special chemicals called, that's right, these are our amino acids. So tRNAs, they have specific amino acids attached to them. And a tRNA has a specific anticodon sequence that relates to a specific amino acid. And thankfully, you don't have to memorize them all, but there's a fancy little table that we can use to help translate the amino acid sequences that we have into protein sequences or amino acid sequences that constitute polypeptides that we call proteins. So the important thing is that tRNAs, they have sequences that we call anticodons. So let's see, our amino acid sequence for the mRNA here, and let's label it, if this is the 5', this is the 3', we know we're gonna have for example, AUG like we said, then we're gonna have a CAA like we said, and let's also add the AAA. Okay, so what's going to happen is that our tRNAs we have a bunch of different kinds of tRNAs, and they all have their own special sequence. This one, for example, could be UUU, and this one, just casually for no specific reason. I'm also going to draw it a bit smaller. Casually for no specific reason. I'm lying. There's a specific reason, but I don't want to reveal it. UAC. And we have another one, let's say is GUU. So these tRNAs, they have RNA, like all throughout their body, and they also have a specific region where they have a set of 3 nucleotides. And what's going to happen is that these tRNAs use those nucleotides, which we call anticodons, and they're going to migrate to the ribosome and kind of bind to the mRNA sequence. And you'll notice that U is complementary to A is complementary to U, and C is complementary to G. That's why these are called anticodons, because they're opposite of the mRNA sequence, which we call codons. And codons are groups of 1, 2, 3 nucleotides that our ribosomes can recognize and ask tRNAs to come in and bind to them. And when these two bind together, that lets the tRNA know that, okay, I can part ways with my little amino acid. And the amino acid will hang around. The tRNA, I knew that was going to happen. The tRNA will, I'm going to have to do this in a weird way. Hold on, let's see if I can just get it to stop being mean to me for a second. Oh my God, of course that happened. That is remarkably unfortunate. We'll be right back. I'm going to have to pause the recording and draw that all over again. So can't even find Zoom. 1 second. Okay, hello, we're back. Sorry about that. So where was I? Oh yeah, so we have our tRNAs and they have specific sequences attached to them that are anticodons. And like we said, these anticodons that they have, what is happening? 1 second. Oh, can you even hear me now? Okay, that should be better. Sorry. So yeah, these anticodons, the tRNAs with anticodons on the bottom that also have this green amino acid attached, what they're going to do is they're going to migrate to our ribosomes, and they're going to stick onto the mRNA, and they're going to bind. And that mRNA is complementary, or the anticodon is complementary to the codon, because we have our codon in blue, and we're going to have the anticodon in black. And if our codon was 5' to 3', our anticodon is antiparallel, so we're going to have that 3' to 5'. We had a problem like that in class, and that was why one of the answers was not correct. We can go over that maybe at the end of this video. So yeah, our anticodons are going to come in, or our tRNAs are going to come in, and the anticodons attached to it are going to bind to the mRNA sequence that is complementary to it. And then once that happens, I detest that. Once that happens, it's going to signal to the tRNA to unbind to that amino acid sequence. And it's going to migrate away. This is villainous. I wish this wasn't happening. 1 second, I'm going to have to figure this out. All right, sorry about that. Still figuring this out. But okay, pretend that the large ribosomal subunit is still there. But what's going to happen when these sequences bind, when they match up, the ribosome is going to recognize that, and it's going to release its amino acid, the little green boy on top, and it's going to migrate away. And it's going to leave behind that amino acid sequence within the RNA. And then another one is going to come in, another one that is complementary to this sequence then, because you know, GUU is complementary to CAA, and then it's going to, once again, it is once again going to detach from its amino acid, and then it is also going to migrate away. And as you see, like the more and more this happens, we're going to be lengthening and lengthening this chain of amino acids. And what is actually going on with these amino acids is that we have these amino acids aren't just like green balls, they are really complicated proteins. And every anticodon is specific to a particular amino acid. And what we do is we have these codon tables. So we're not going to consider the anticodon, we're going to consider the actual codon sequence. So we knew that, so this original UAC here, this anticodon, it was complementary to AUG. And AUG is a very special codon. It was the methionine codon. So if I had to redraw it, which I am going to for some reason, yeah, before it detached, like this was always a methionine. Let me think. Okay. Yep. So these tRNAs that we have and the anticodons that they have attached to them, or the anticodons that are part of them, they are attached to a specific amino acid, but we don't use the anticodons to like describe the amino acid that they're attached to. We actually use the codons that the anticodons are complementary to. So we can look at the original mRNA codon sequence, and then we look at a codon table to see what amino acid that is eventually going to come with, and it's eventually going to build a long chain. So when we're looking at these codon charts, we're considering the mRNA sequence, not the anticodon sequence that is a part of the tRNA, which actually delivers the amino acid. We're always worried about the mRNA sequence. And that makes it easier on us because we have our mRNA sequence here, and we don't then have to translate it into an anticodon sequence to translate it into an amino acid sequence. We can just look at our nice mRNA sequence, go to a codon table, and find, oh, AUG, that means it's going to be a methionine. And so we know that we have a methionine here. Same thing, we have CAA. We can go to our table and we find CAA right here. We know that's going to make glutamine. Oh, and the way that you read these tables is, so let's say you have the codon GUC. The first codon, or the first nucleotide base in this codon is a G. And so we go to the left side with the first base and we see, oh, G is here. The second codon is U. So we look at this top side and we say, oh, U is here. So we go here and here, kind of like if we were doing a Punnett square. We connect all the dots and then we see that the third one is C. So we can look in here and find GUC. It wouldn't matter because we have valine, they're all valine, whether that's GUU, GUC, GUA, or GUG, and that'll actually be important in just a little bit. Yeah, we can look at our mRNA sequence, which we already found and described, and we can then look at a codon table and translate it. So AAA, we're going to have lysine. We had our methionine, our glutamine, I think it was, yeah. Methionine, glutamine, and then AAA was lysine. GUG, GUG is once again valine. And UGA is a special one. UGA stop. And that'll come in handy for when we want to end this, but just for now, let's call it a stop. And this is the amino acid sequence that results from this mRNA sequence. So yeah, we've just made an amino acid sequence via translation. And this process here is a bit more complicated than I drew, but not too much complicated, not too much more complicated. And we'll talk about it in just a second. Okay, so we did that, and our sequence is methionine, glutamine, lysine, valine, and a stop codon. So part B, how would our amino acid polymer change if there is a deletion at nucleotide 3? What kind of sequence mutations would this be? Okay, so amino acid polymer. What's a polymer? We can break it down. We've heard this word before, I'm sure, but mer means unit or units. For example, if you're making a tower of Legos, I guess, the individual units that make up that tower is just a single Lego brick. If you have a carbohydrate, the unit is probably a glucose molecule or a fructose molecule or a lactose molecule. So the mer are the individual units, and poly means many. So a polymer is just something that's made of many units, kind of like an amino acid sequence maybe. Also, DNA is a polymer. It's made of many units of nucleotide bases. So our amino acid polymer is simply a long chain of amino acids. How would our amino acid polymer change if there is a deletion at nucleotide 3? Okay, so deletion at nucleotide 3, we can just look at our sequence up here and say, okay, nucleotide 3. So we have 1, 2, 3, 1, 2, 3. This one here, actually, yeah. And the third one gets deleted. Third one gets deleted. Third one gets deleted. How is this going to affect our amino acid sequence? So remember, our tRNAs are always in anticodons of 3. And they're going to attach. So if this was deleted, then what's going to happen is that we're not going to have this particular strain of mRNA. If the G was deleted, then it's like the frame in which we're looking gets shifted. Because, we're always, our enzymes are reading in pairs of 3. So we're no longer reading ATG, CAA, we're reading ATC, AAA, AAG. And so our codons, instead of being this way, they kind of shifted a little bit. And we call that specific mutation a frame shift mutation, because we're shifting the frame in which we read, because we always read codons in groups of 3. Or we read our nucleotide bases in groups of 3, in groups that we call codons. So this kind of sequence mutation would be a frame shift. but it's a specific kind, and we'll see why. So we have our frame shift mutation. And what this is going to do is that we no longer have that, we no longer have that AUG, we have an AUC. And so the tRNA that's going to come in is going to be a UAG with a specific amino acid attached to it. But what do we know about translation? For all these processes that we're doing, we know replication, transcription, and translation. They have 3 steps, which are initiation, elongation, and termination. So how is translation even initiated? Well, we know that our small ribosomal subunits bind to our mRNA sequence, and then the large unit binds on top of that. But what do we really need to start doing in order to create an elongated sequence of our amino acids? We need to have a specific codon start the process. And that's why methionine is so important. We call it the start codon. All amino acid sequences have to start with methionine because that is how our enzymes know that this is a new strand of mRNA, and we're going to make a functional protein off of this because we know we're starting at a specific spot and we don't want to mess up our protein. So we're going to start at a specific spot, a specific sequence that has to be AUG for our mRNA. And that's going to give us a specific amino acid, methionine, that we can then build off of. So if we had a deletion at that 3rd nucleotide, we no longer have ATG becoming TAC becoming AUG. We now have ATC in our complementary strand, that is a TAG. Hold on one second. We no longer have a complementary ATG. We have an ATC. ATC, because this 3rd one here got deleted, and that became TAG in our template strand. And then our mRNA would therefore be AUC. And if we have an AUC here, what is that going to code for? It's going to code for isoleucine. And that is not a proper start codon. So without that original start codon, we're not going to have anywhere to start. So we're not even going to translate the rest of this sequence because we don't have that original start codon. So we're going to have no amino acid sequence. That's a 0, not the circles I've been drawing. So this is a special kind of mutation called a nonsense. Wait, is that right? Hold on, let me think about it. I think that's right. A nonsense mutation is either when you don't have a start codon or you don't have a stop codon. I'm pretty sure it's a nonsense mutation. So it's a frame shift, but specifically a nonsense mutation because we're not sensing the proper codon to start, and therefore we don't have the adequate anticodon to attach a methionine to. And without that methionine, we can't start the process of elongating a sequence of amino acids. So we're not sensing the right codon, we're sensing an AUC instead of an AUG, and therefore we don't build any amino acid sequence. So how would our amino acid polymer change if there's a deletion at nucleotide 3? Because we're losing out on this AUG, we just wouldn't build it at all. That's the change. The change is that we go from MET, GLN, LYS, VAL stop to nothing at all. So we would be prevented from making a proper amino acid sequence. Okay, part C. What would the product be if codon 3 came from its own exon? Let me clean this up a little bit. if codon 3 came from its own exon, okay, sorry, I can't think and write at the same time. It would be the product if codon 3 came from its own exon and wasn't a part of this sequence. So we know from a certain process, we have to process our RNA. It doesn't always come out as this nice mature RNA. It first is transcribed into immature RNA. We have to cut out some sequences, attach some other sequences together, add some protective measures to our mRNA strand, like our GCAP and a poly A tail that we mentioned earlier. And once again, this isn't a review of transcription, but we know that DNA is typically made up of sequences that we keep that go into our mature RNA and sequences that we cut out. And we do this by way of splicing. And what our spliceosomes do, they're actually really complicated. They're made up of RNAs called SNRPs, which is small nuclear ribosomal something, polymerases, or something like that. You don't have to know the name, but they cut out these introns. Oops, that shouldn't have been an eraser. They cut out these introns, and then we have nice exon sequences that these SNRPs then stick together. And these exon sequences are actually made up of nucleotide units. These intron sequences were also made up of nucleotide, but they weren't the kind that we use to actually make specific amino acids. They would have made amino acids, but we don't want these amino acids. We want to make a specific product. For example, we want to make hemoglobin that's made up of this combination of amino acids. And if these were in there, then we would make a longer chain that wouldn't actually be hemoglobin. So we typically splice our DNA, and splicing is cutting out the stuff we don't want and then reattaching the stuff that we do want. So like we're slicing, but putting it back together in the way that we want it. And so if that was the case, and let's say that every exon was just a single codon. So let's say that these were each their own exons. So this was an exon, this was an exon, and so on and so forth. Let's say that codon 3, and remember codons are groups of 3 individual nucleotides. So we have one codon here, one here, one here, one here, one here, because they're all made up of 3 nucleotides. So let's say codon 3 here was its own exon. And let's say that this wasn't what was spliced together, because we have our exons, and usually they're separated by introns, but we cut out the introns and attach the exons together. What if we did that, but we left out one of the exons? We also cut out one of the exons and just attached these 2 exons together. This used to be an exon that we included in the sequence, but we cut it out and didn't include it in the sequence of exons that we are then going to turn into an amino acid sequence. So we're still splicing, but we can do it in a different way. And that is the second part of this question. What process could make this possible? A process called alternative splicing that we investigated in previous classes. I'm not sure if I'm going to make a review of transcription. I should, because it's really complicated, but not today. So alternative splicing, we were able to not include that third exon. So let's say that we didn't include it. Didn't include it, didn't include it. What would be the product if codon 3 came from its own exon? So if we just didn't include this sequence, are we shifting the frame that we read our codons in? No, because we read in pairs of 3 and these codons are still intact. We're still reading 1, 2, 3, 1, 2, 3, 1, 2, 3, and 1, 2, 3. We just happen to have cut these 3 out. So we're no longer making that lysine, but the rest of it should be preserved. We're still going to turn AUG into MET. We're still going to turn CAA into our glutamine, still GUG into our valine, and UGA into our stop codon. So what would be the product? Let's see if we had to write it all back out again. AUG mRNA would be methionine, CAA, glutamine, GUG, our valine, and UGA, our stop codon. So we'd have this as our product. Okay, part D. Sorry, that was a bit inarticulate, but whatever. It's okay. I'm doing a great job. I believe in myself. So part D, if codon 4 experienced wobble, that's a funny word. If codon 4 experienced wobble, what is the most likely difference that would result in our amino acid polymer? So what is wobble, first of all? I'm so glad you asked, Gareth. Wobble is a quirk of our mRNA sequence. So we have our mRNA sequence, and it is attached to that small ribosomal subunit. And it has its own sequence, AUG, TAA, AAA, GUG, and UGA. So we know that A typically pairs with T. Actually, that's not true. In RNA, A typically pairs with U. We know that G typically pairs with C. And we know that our tRNAs have specific anticodons that associate with specific amino acids. For example, let's say that, I need to erase this so I can actually see. Okay, so let's say that this guy here is AAA. We know that the mRNA sequence AAA will eventually create or have an amino acid known as lysine be polymerized in our ribosomes. And that is because of the binding of the anticodon UUU. But what if maybe we had a mutation and this A had been mutated into, I used the pencil, of course. If this A had been mutated into a G instead. So we didn't shift our frame. We had a single nucleotide mutation, which we call a single nucleotide polymorphism. But we know that we needed this lysine here. Could lysine still like come in and do the thing? It could actually, because of a concept called wobble. And the thing is, we can't forget that these are all like actual chemicals. And they exist in physical, like three-dimensional space. Why isn't this working? Okay, they exist in three-dimensional space. And this G is going to kind of, actually, that's not true, but we know that A and U pair together nicely. U and G do not pair together nicely. They, they don't fit together. G typically creates 3 hydrogen bonds with C, and A and U create 2. So G is used to having a partner with 3, U is used to having a partner with 2 of these hydrogen bonds. So if U comes together with G, they have different expectations and they don't fit together nicely like they should. But remembering that these are chemicals that exist in three-dimensional space, what can happen, one of the very fortunate things that can happen is that the U gets moved out of the way a little bit. It's not like in the space where we expect it to be. It kind of shifts in its seat on the anticodon a little bit. It, wobbles a little bit out of the way because it has an unexpected number of hydrogen bonds to deal with. And this wobble property, the ability of, this tRNA will still bind. It will still recognize that, yep, this is where I need to drop off my lysine, and we're good. We're still going to add lysine into our sequence. And then our tRNA is still going to move up out of the way, and everything will be wonderful. We're still going to have added our sequence. And the fact that this was a G instead of an A, it doesn't affect the overall sequence that we had. And this property of DNA is called wobble. And this can only happen at the 3rd position, because if you think of it like a lock and key, we have a lock that has specific set of metal pins in it, and we have a key that has a corresponding number of teeth on it. What's important is that, let's say you need a Bluetooth to get in there, and you need a red tooth to get that far in, and you need a yellow tooth to get this far in. As you get further and further in, oops, I didn't think about that. So the Bluetooth will hit first, and we know that, okay, we needed the blue one, then the red one, but oh, the yellow one doesn't fit in. Oh wait, damn it, I messed that up, sorry. Let's try that one more time. I didn't prepare this beforehand. So we had a blue tooth, and then a red tooth, and then a yellow tooth is what you need to usually get into this lock. But we had a blue tooth here, and then a red tooth here, and then a green tooth here, even though the pen isn't letting me write. So we go in, this is a better thing. We have our blue tooth matching, and then our red tooth matching. Then when we get here, we don't have a match. But the key is still far enough in, pretend that the key is still far enough in that we can get to the end and unlock the door. So the further in we go, the further in we already are, the less important it is that this 3rd mer, this 3rd unit from our polymer, that it matches what we needed to get in. It's less sensitive. This 3rd nucleotide is less important for our tRNA's ability to bind our anticodons to it. That was a terrible explanation, but it permits this wobble effect that allows us to still insert the proper amino acid into our amino acid polymer that we need to make specific proteins. So if codon 4 exhibited wobble, what is the most likely difference that would result in our amino acid polymer? I kind of mentioned it earlier, but it is most likely that, so because of this difference in the number of hydrogen bonds that our nucleotide bases usually have with its complement, that affects how likely wobble is to occur. So in our AAG, since G is so similar to C, actually no, let me rephrase that. I'm not surprised that this is happening. Anyway. So AAA, we usually have an A here, and that usually creates 2 hydrogen bonds with its anticodon. And that's good. That's what our anticodon expects, and that will more easily enable it to bind, and then that'll signal to our tRNA to release its amino acid that's attached. And this expectation of 2 hydrogen bonds means that the other one that produces 2 hydrogen bonds will also kind of still have that expected number of hydrogen bonds. So the U would have, like there would still be wobble because, U doesn't bind with U, A binds with U. But the fact that this U, if it were to result in this place during a mutation, because it still has those 2 hydrogen bonds, it would fit in a little bit better, and the wobble would be less intense, and it would be even more likely that this chemical reaction that allows our anticodon to bind to our codon, it would allow that more likely. Like, it can kind of fit in and disguise itself. It still has those 2 hydrogen bonds, but a G, kind of like we said earlier, because it has a different number of hydrogen bonds, it would create a more intense wobble. Like the U would be kind of pushed up here. And what that would actually look like is that our entire tRNA would be a little off balance. And if it doesn't like click into place, then tRNA isn't going to get the signal to release its anticodon, and we're not going to be able to keep transcribing, or we're not going to be able to add this particular sequence, this particular amino acid. So if the wobble is too intense because you have a mismatch in the number of hydrogen bonds that your nucleotides are expecting, that your tRNA really needs, then you might not be able to polymerize that amino acid sequence. The wobble could be too intense because the nucleotide bases are too incompatible. But like getting back to the actual question, if codon 4 exhibited wobble, let's investigate that. So we know our codons are number 1, 2, 3, 4, and 5. So let's focus on number 4. If codon 4 exhibited wobble, what would be the most likely difference that would result in our amino acid polymer? So we have a sequence GUG. We know wobble can't happen in the first or second nucleotides because our tRNA anticodons really need those first 2. Kind of remember with that lock and key example. So they really need that the first 2 nucleotides to be the perfect matches. So G has to have a complementary, why isn't the pen working? G has to have a complementary cytosine, and U has to have a complementary adenine. But G would prefer to have a C, but if we experienced wobble, like the question is asking, what nucleotide would be the most acceptable, the least likely to create that problem where our tRNA is just so off balance that it doesn't get the signal to release our guide, where it could be more on balance so that it can get the signal to polymerize our amino acid sequence. So we would prefer a C, and we know that cytosine usually has a 3 hydrogen bond thing going on with guanine. So guanine would have the most appropriate, or sorry, this is wrong. This like kind of has to be the C because that's what exists. But if we had a mutation, I think you get the point, even though I messed up. So we would typically have a CAC anticodon. And what nucleotide would most likely fit in this third spot if it wasn't a G? And that would be a C, because we know our anticodon does have this CAC sequence, and the codon, therefore, would preferably be GUG. But if we had wobble here, and this, instead of being a G, it was some other nucleotide, they would be most compatible if they maintained this 3 hydrogen bond business going on. And the only other nucleotide that has 3 hydrogen bonds aside the G that we would like to have is C. So our CAC anticodon would like to bind, or could bind to a GUC, and it would still be like straight enough. It would still have a minimal wobble that would allow it to polymerize the amino acid sequence. Sorry, I did not explain that well. And if we had GUC as our mRNA sequence, that's going to code for a different amino acid probably. So let's look. We wanted GUG, but we had GUC. Oh, wow. Very fortunately, that still codes for valine. That's very good, very nice. So even though we had this mutation, we actually technically wouldn't experience wobble. I should have done a better example. I should have prepared this beforehand. Whatever, you get the point. A better example would be like if we had this business, where the sequence we originally had was CAC, but then we had a mutation that this was no longer a C, it was instead a G. That would that could mess up, but because we know that we wanted this histidine here, we could still have our wobble, and it would end up giving us our histidine, even though this was GUC. Sorry, I dunked that up, but whatever. If you have any questions, please let me know. I made this up. I made this question in like 3 minutes, so it's not the best. But actually, while this recording is going, let's see what is actually happening. So this is an animation that kind of helps to visualize what's going on with translation. So we have this orange strand of stuff on the bottom. We have that orange strand of stuff on the bottom, and there's little teeth coming off of it. Wait, I think I gave it away. This orange strand is our mRNA strand, like we were drawing in blue in the picture that we have been doing for a long time. And we have the small ribosomal subunit that it's sitting on top of. I should probably switch colors. I'm being a menace, I'm sorry. So we have our orange, it's not really orange, it's more like a yellow. We have our yellow sequence. We have our nice light blue small ribosomal subunit. Then we have our large ribosomal subunit. And then we have, okay, so we have the teeth on this RNA, this mRNA. And we know that mRNA is made up of, RNA nucleotide bases. And when a ribosome fits on top of it, a ribosome has a specific anatomy and some special helpers. So what's going to happen is our tRNAs, like we were talking about earlier, they have their 3 sequence anticodons. And those, please let me, they are going to move in and bind. So our anticodon will bind to our codon. Oh, and I also forgot that they have their amino acid attached. But there's also, other anticodons floating around in our cells. Duplicate this. Can I duplicate this? Yes, copy. Let me paste it, please. No luck. Okay. That's going to do its thing. It's going to jump in. Of course, it's not attached. It's going to jump in at a certain point. Okay, this isn't working, so I'm just going to look at the picture and describe it. It's going to jump in at a certain point. And then it's going to, once it binds here, it's going to get the signal and it's going to cut off its amino acid. And then another one is going to jump in. Actually, I'm just going to show a video because I'm tired of talking. 1 second. Oh God, why is this happening? I wanted a shortcut, but it's not working. That's better, except it's just not going to work. So I'm going to stop. And we're going to watch this and have a Englishman explain it to us.
To produce a functional protein, the genetic information found in the form of an mRNA, which is composed of nucleotide building blocks, must be translated into the language of proteins, which are composed of amino acids. This translation process is orchestrated by a large macromolecular ribonucleoprotein machine in the cell, known as the ribosome. It uses a key adapter species known as a transfer RNA or tRNA. tRNAs are the molecules that actually translate the nucleotide sequence into an amino acid sequence. tRNAs are bifunctional in nature, with 3 nucleotides poised at one end of the molecule to read the mRNA template in an anti-parallel fashion. This is referred to as the anticodon end of the tRNA. At the other end of the molecule is the acceptor end, where an activated amino acid is attached. This amino acid corresponds specifically to the identity of the codon being read. Aminoacyl tRNA synthetase enzymes are responsible for catalyzing the loading of the amino acid onto the appropriate tRNA species. The fidelity of this step is extremely high, which is critical to the overall fidelity of protein synthesis.
The process of elongation for now.
In its most basic form, during translation, the tRNAs walk along an mRNA in the 5' to 3' direction. As they move, the tRNAs sequentially recognize or decode consecutive 3 nucleotide codons that correspond to a given amino acid. This highly orchestrated march along the mRNA template takes place in the heart of the ribosome, facilitated at each step by portions of this machine, as well as by extracellular factors that participate in each step during the translational cycle. The ribosome has binding sites for 3 different tRNAs. One tRNA is always bound in the centralmost position in the ribosome and carries the growing polypeptide chain. We refer to this tRNA as the peptidyl tRNA. It's bound in the P site of the ribosome. The incoming tRNA with the new amino acid to be incorporated binds adjacent to the P site in a site referred to as the A site for aminoacyl tRNA. There is a third binding site, the E or exit site, where the tRNA binds on its way out of the ribosome following the incorporation of its amino acid into the polypeptide chain. As in most processes, there is a beginning, a middle, and an end. The initiation phase of translation consists of the small subunit of the ribosome working in concert with various initiation factors to identify the starting AUG methionine codon that is positioned in the P site of the ribosome. Next, the initiator tRNA engages the AUG codon and a large subunit of the ribosome joins in a catalyzed reaction. Because of the substantial differences in the mRNA structure between bacteria and eukaryotes, this process is quite different in these two systems.
A long video.
In bacteria,
really.
a polypurine motif in the mRNA, known as the Shine-Dalgarno motif, is found in the capped end codon. Because the initiator tRNA is part of this scanning complex, it plays a key role in finding the AUG and positioning the ribosome complex where it belongs.
So remember to initiate our translation.
After
we
initiation
need to
is
find
complete,
that AUG,
the ribosome is ready for iterated rounds
codon.
of amino acid addition. This
and
phase,
once we find
referred
the
to as
start,
elongation,
then we can just
be broken down into 3
polymerize our
basic
amino acid
steps.
sequence like we intend
In the
to.
first step, the ribosome must select the aminoacyl tRNA that corresponds to the codon poised in the A site of the ribosome. This step is facilitated by the GTPase elongation factor EFTU in bacteria or EEF1A in eukaryotes. Simply put, the process of tRNA selection consists of 2 phases. In the initial selection phase, an aminoacyl tRNA in complex with EFTU can be rejected or accepted by the ribosome.
tRNA with
Once the complex has been accepted,
amino
GTP is
acid that we are
hydrolyzed and
looking for
EFTU leaves the
just like
ribosome.
migrates
The
into
near-cognate
the
tRNA can still
ribosomal
be rejected in the second phase,
subunits,
which is referred to as
it's a bit more
proofreading.
complicated than that.
In both steps,
we
thermodynamic
actually
and
have
kinetic mechanisms are
some
used by the
molecular
ribosome to aid
mechanisms
in
that
discrimination, such that the system chooses
like
with extremely high fidelity the cognate tRNA
what
from the 30 or 40
tRNAs actually enter
tRNAs in the cell.
ribosomal subunit.
Once the tRNA selection step has taken place, peptide bond formation is catalyzed in the large subunit. In this step, the growing polypeptide chain is transferred from the peptidyl tRNA to the nucleophilic amine of the A site tRNA amino acid.
And this is what I've been saying,
extends
that
the polypeptide chain
when we
by 1 amino
recognize,
acid.
or when the tRNA
At this stage, the
binds
extended
properly
peptide is attached to the tRNA in the
corresponding codon sequence. So we have our mRNA codons in purple at the bottom, and then those big orange blobs are our tRNAs, and we have the anticodons that are binding to the purple sequence. And when that happens, I've been saying that like signals to the tRNA to release the amino acid in green at the top there, but what it's actually doing is transferring it to the tRNA before it. And when that happens, we to stick amino acids together, they create what are those called? Peptide bonds, sorry. And you can see that one by one, we will
this
be
whole
adding
mRNA
on
tRNA complex must now be
a single
repositioned in the
amino acid
ribosome
to
open
an
up the A
ever-growing
site for the
chain.
next.
But as it articulated, the way this happens is that we have in our ribosomes, we have different sites, different parts. We call them the E site, P site, and A site. So our tRNAs will come in. One of our guys sits on it in the P site specifically, and it has its little guy attached. So first, a new tRNA will come in and it will sit in the A site. And I think of, or I remember that it first sits on the A site because, A is the first letter in the alphabet. So a new RNA, a new tRNA will start with the alphabet, the first letter in the alphabet. And then when it binds to that codon sequence for which it has the appropriate anticodon, that will tell this tRNA that it's time to polymerize. And that's why it's called the P site. It is where we polymerize. And remember, polymer is to make many units of something. So to polymerize is to extend that polymer. So if we polymerize, we're going to add another one on to extend the chain even further. And that's what's going to happen. This little guy, or I guess this little guy is going to click attached to this, and that is going to tell this tRNA to move to the E site, and it will also get this tRNA to move from the A site to the P site. So think of this as, again, it's hard to just draw or look at it in a picture because it's a moving process. We're going to be moving this way. Like all of our stuff is constantly moving this way, this way, this way. So we started with our new guy coming into the A site. There was a guy in the P site. And once a tRNA gets to the E site is the exit site. It's where a tRNA will get up and leave. And then we can start the process over again. We have a new tRNA with a new guy, and it will come in to the A site, because remember, it starts at the first letter of the alphabet. And once we have that bonding of the anticodon to the codon, that signals to this guy, this tRNA in the P site, that it's time to polymerize. And then the process just keeps going. This guy will move from the P site to the E site. A site will move to the P site. And then once again, a new tRNA is recruited to bring a proper corresponding amino acid that we want for a specific protein comes to the A site. Whatever. And that's what we see here in this animation. We see a new one, a new tRNA coming into the right side there, sticking to the A site. And then we have a guy already in the P site in the center. And once the guy binds to the A site, the guy in the P site will stick its long chain of red amino acids onto the one in the A site. That will polymerize the chain of amino acids. And then they will all shift to the left so that the person who was in this P site goes to the E or the exit site. Then the one who was in the A site will go to the P site. And that is pretty much all of translation. It's kind of complicated, but hopefully this was, I know I babbled a lot and I wasn't really clear at every point. So if you have any questions, please do let me know. And yeah, thanks for watching. Hope you're feeling it, having a wonderful day. And I will see you in class on Thursday.